I can see your screen now. Wait.
Okay, can you hear me okay?
Yeah, you can share the screen.
All right, let me share my screen. Number two. Okay, I'm good.
Yeah. Are you in hotel?
Yes, I am.
Okay.
So I'm not quite sure how the internet works, so I'm gonna keep the video off just to save the bandwidth if it's okay.
Yeah, yeah, sure.
How are things, Al?
Well, yeah, I mean, I think our branch chief is in the hibernation mode.
Okay, that's safe.
Yeah. When are you coming back?
I'm coming back on 5th, the day after tomorrow. So I see you
Saturday.
Saturday, yes. I have a event to attend in DC on Saturday afternoon. How's the conference policy now? Are we still not allowed to?
No conference.
No conference, so this is illegal.
This is okay. I mean, online the the Zoom is okay. It's just no conference, no travel conference.
So if I go to DC for event, local events, that should be fine, right?
Yeah.
Yeah. Okay.
Uh, shall we get started?
Sure. Go ahead.
Okay. Okay, so let's get started. So hello everyone. Welcome to our today's Zoom seminar. So it's really my pleasure to welcome my friend and my also colleague, long-term collaborator, Dr. Wei Li from National Eye Institute. So Wei currently is chief of retina neurophysiology section. Wei finished his bachelor degree in Zhejiang University, China, and then move on for the graduate training in neuroscience in University of Texas, Houston Health Science Center. And after which, he continued to pursue his postdoc training in Department of Ophthalmology in Northwestern University School of Medicine. So since 2007, when started his research program in National Eye Institute to this very day. So he's currently, as I mentioned, as a senior investigator, also the chief of retina neurobiology section in the NEI, NIH. So, from the title, I think everyone can see that we're actually interested in some very peculiar animal model, which is 13 line ground squirrel. Actually, part of his research is focused on hibernation. So, his lab actually mainly interested in the mammalian retina as a model for the central nervous system to understand how it functions in physiological conditions. His lab has focused on mainly two research directions. First of all is photoreceptor structure, synapse circuits, and then development. Um, and second part of his research program is hibernation and metabolic adaptation in the retina and beyond. So, as I mentioned that beyond the neuroscience, part of Wade's lab also interested in working in neuroimmune metabolism during hibernation, using this very unique animal model, 13-line ground squirrel, as you can see on the on his front page. And then they are particular focus on microglial biology, hoping to discover new therapeutic strategies for neurodegenerative diseases. So, personally, over the years of collaboration, I have greatly admired Wei's lab and his insight and storytelling in project development, as well as the ability to integrate different disciplines into his work, which is truly impressive. I believe everyone today online will really enjoy Wei's lecture. So please go ahead, Wei. We are looking forward to your talk.
Well, thank you, Tran. Thanks much for the invitation. And as Tran said, we has been long-term friends, collaborators at NIH, and I have been uh, a backup host for this immune Zoom series once when Tran is not available, and now I am the backup speaker. This is what friends do for each other, being the backup for each other. So it's a great honor to be on Immune Zoom again. So as Sean mentioned, I mostly study retinal physiology, and just so happen I use this animal model called 13-line ground squirrel, and just so happen this is a mammalian hibernator. So it got me interested when I started my own lab to look into research project not necessarily related to vision, but as you can see today, it will come back to the visual system, the retina that I've been using for as model for many years. Um, so just a brief introduction of hibernation. I think it didn't come across in this immune Zoom talks. And again, I'm not immunologist, so I really come across this topic of new metabolic immune regulation by chance, and I'm here to look for advices and suggestions from this community. So hibernation is a fascinating biological phenomena many of you know, and usually, but not always, in the wintertime when animals don't have a lot of food availability, some of them evolve to solve the problem by bury themselves on the ground and without eating and drinking for a prolonged period of time and regulate their body temperature very low, just a couple of degrees above ambient temperature. The animal model again we use called 13-line ground squirrel. It's a rat size small mammal. As you can see, their normal body temperature is around 35, 37. But in the wintertime, they will drop their body temperature down to 4 or 5, just a couple of degrees above environment. But they don't always stay at this low temperature. In fact, they have this periodic swing back to the normal body temperature, so-called interval arousal from time to time, as you can see. And those drastic change in body temperature, metabolism, and circulation could be also detrimental to a non-hibernator. But through this period of winter hibernation, these animals will not have any cold damage due to the low body temperature. imagine yourself, unfortunately, lock yourself in a cold room when you're doing an experiment and nobody finds you until a couple of days later, you'll be dead. But those animals survive low temperature for 4 or 5 months without tissue or organ damage. And again, as I mentioned, those interval arousal involve very drastic circulation, metabolism, body temperature change, uh, a condition somewhat akin to ischemic reperfusion. But again, there's no injury associated with those transitions. And very obviously, if you say lay in a bed for 4 or 5 months due to injury, then your muscle and bone will have dystrophy. But none of those happens to those mammals. So if we can understand a little bit of the biology of hibernation, they could have a lot of medical clinical applications, as you could imagine. In a case of a critical care, stroke, and cardiac arrest, also applications for tissue organ preservation before transplantation. If you think about it, when a tissue or organ are harvested before transplantation, the storage condition is very similar to hibernation. There's no oxygen supply, but they're at a very low temperature. So how to prolong this period is a critical need, medical need. And also, there's other cellular protections that could be used in the case of an injury. And for today's talk, I'm going to mostly focus on neuroprotection. So, in order to study the cellular molecular mechanism of co-adaptation or co-protection, several years ago, our lab, particularly then postdoc Jingxin O, developed an iPS cell from this particular species, the 13-line ground squirrel. And we use this as a tool to compare cells from ground squirrel versus a non-hibernator, for instance, human cells, and we identify some of the cellular molecular mechanism that allows the ground squirrel cells to survive cold temperature for a long period of time. And I'm just going to briefly mention one thing, one mechanism, that is the avoidance of mitochondria rust overproduction that we identified as a key cellular mechanism for cold adaptation. And I'm going to come back to that point. And with this platform of iPS cell, we further did screening, and in collaboration with Jingxing, who is now very successful with his own lab in Guangzhou, we identified a more general co-adaptive mechanism involving the nuclear translocation of a transcription factor called FOXO1. And that being a very generic co-protective mechanism. But along the way, we've been always thinking whether those animals and the cells from those animals could be more resilient to stress or challenging other than cold temperature. Because if you imagine, if we wanted to apply to clinical situation, low temperature is not always the best option, right? Particularly the whole body temperature, we cannot put patients in need at low temperature for a long period of time. So we've been thinking about this, whether the ground squirrel cells and tissues could be more resilient to other insults than cold. And being a retina physiologist, we think more of the central nervous system, of course. And in fact, hibernation has been touted as a model for neuroprotection for quite some time, and in fact, a couple of decades ago, because of its ability to survive very difficult conditions. So what we are hoping to achieve is some mechanism or strategy that is related or originated from hibernation, but eventually could be co-independent for the application of neuroprotection and other cellular protection. So to this end, we had a very close collaboration with Josh Benstock and Luca Perizzotti Hameli, both at Cambridge, one is Harvard, one at Cambridge, UK. Um, and we performed a unsupervised metabolic and transcriptional profiling of hibernating ground squirrel, taking samples from different phases of hibernation while they're awake, when they're entering hibernation and they're deep in torpor and they are rising from the torpor into interval. So different time point and perform metabolic and transcriptional analysis. So we did that for the brain and also cross-validated with retina. So when we do the metabolic analysis, it turns out different phases of hibernation samples can be fairly well separated, as you can see here, and by many metabolic pathways. And the pathway enrichment analysis through that we identified a total of 19 enriched clusters, and with number 7 showing the highest combined statistical significance and weighted metabolite scores. And pathway analysis of this cluster 7 showing a lot of metabolites in the glycolysis and TCA cycle. And specifically, if you look at the TCA cycle, we found that metabolites before the transition of succinate to fumarate being upregulated during hibernation, or more have more in the hibernation, while the metabolites below the transition of succinate to fumarate uh, have less during hibernation. So indicating a potential break of TCA cycle right at this transition of succinate to fumarate. And simultaneously, the transcriptional analysis also show well separated stages of hibernation. And among those DEGs, we also identified many associated with succinate dehydrogenase. To correlate those metabolic findings and their associated transcriptional changes, we performed a analysis of DGs with known metabolites function associated with this cluster 7, and using this enzyme commission numbers associated with each significant genes and metabolites. By doing so, we identified many unique enzymatic functions, out of which only 7 are shared by both gene and metabolites when compared the awake versus hibernating squirrels. And 4 of the 7 maps to eventually converge to the succinate dehydrogenase and succinate metabolism. So we did a very similar analysis from the separate data set from retina, and had a very similar result. And we, in fact, can combine 2 data sets together. And again, succinate dehydrogenase is coming on the top as the most significantly changed molecule, indicating that this potentially could be a key regulating point for tissues from central nervous system during hibernation. And when we, in fact, biochemically measured the succinate dehydrogenase directly, we can see that its activity is lower with normalized condition in hibernating retina. And we had the same, very similar result for the brain tissue. So in the condition of hibernation, we through unsupervised analysis identified succinate dehydrogenase as the key regulatory point. And but does hibernation indeed confer neuroprotection? For that, we need a good model. We wanted to know how do hibernating ground squirrel respond to injury, particularly neural injury. To this end, we developed this axonal injury model. It is a very classic model called optic nerve crush, which is an injury of the optic nerve. It's composed of axons from retina ganglion cell in the retina that projecting to the brain. And this is a very classic model for neuroinjury. But we have developed two novel aspects of this model. One is that we are able to develop a way to injury only half of the optic nerve so that in the same tissue that we will have a injury site versus uninjured site to compare with. And this is taking advantage of a unique optic nerve structure from this animal compared to so they come out of the eyeball in a linear fashion rather than the traditional model. For instance, mouse, the optic nerve come out and optic nerve head at a particular point. So this linear structure allows us to injury half of the optic nerve, which, as you will see, become very handy. And second, we were able to develop surgery methods such that we can perform the surgery during hibernation without waking the animal from hibernation. So first, when we do the injury in the awake animal, as you can see that uh, uh, from the injury site, the retina ganglion cell will die due to this axonal injury. And this is a antibody labels retina ganglion cell. On the flip side, the uninjured part, the retina ganglion cell survived very well. And as you can see, there's a sort of boundary between injury and uninjured site. If we zoom in, you can see clearly that this is 2 weeks after injury. There's massive retina ganglion cell death from the injury site. But when we perform the same injury in the hibernating animal 2 weeks later, you can see that neurons survived very well without much of a cell death. And this is a quantification. As you can see, the surviving curve for the awake animal is down to about 20% for 3 weeks, whereas the hibernating animal, the neurons survived the axon injury. Not only that, we can measure their activity physiologically using a multi-electro array, showing that those neurons are not just dead bodies, have not been cleared because of hibernation. And multi-electro array activity shows spontaneous activity is really low in the awake injury model due to cell death, but in the hibernating model, the activity maintained. And this is just quantification. So on one side, we saw succinate dehydrogenase inhibition during hibernation. On the other hand, we saw neuroprotection, but we do not know whether they're linked and whether what was the what could be the mechanism for neuroprotection. So to this end, we take advantage of the fact that we have the injured and uninjured tissue of cells from the same tissue. So we can do a bulk RNA-seq from retinas of injury on injury site. And we saw that for the awake animal, there are a lot of genes go up and down due to injury. But this transcriptional response is very much muted in the hibernating conditions. And among those DEGs, we identified pathways related to inflammation and immune response and cytokine and so on and so forth. More detailed DEG analysis also identify a lot of genes associated with microglial macrophage, for instance, C1q, CD68, TREM2, etc. So that really led us from a neurocentric view to the other side, looking at microglia and macrophage. First, we're looking at the injury site. So let me orientate you. This is optic nerve, and retina will be far away, and we inject a dye called CTB, and those can be transported through axon from the retina to the brain. As you can see, because of the crash injury, the transportation has been stopped here. So in the awake animal samples, as you can see, the injury site has a large accumulation of IBO1 positive microglia or macrophage. But this is totally devoided in the hibernating animal. At the injury site, you have literally an area that doesn't have a lot of cell at all. Uh, there's no obvious aggregation of microglia. So this is a very big difference. Now, this is not too surprised because microglia are local immune cells. They respond to injury. So, but the cell death really happens miles away in the retina. So we decide to look at the retina, what happens there. So this is a cross section of the retina. As you can see, the microglia labeled with antibody against IPA1 here in green resides in the synaptic layer. And when we focus the microglia in the retina ganglion cell, where those dying neuron resides, if we look down in a whole mount view, we can see for the awake animal, as time goes by after injury, neurons progressively dying, and you can see a cumulative microglia aggregation, and they become more and more active, indicated by the CD68 staining. But the picture is very different for the hibernating animal. 3 weeks into hibernation, as you can see, the neurons are survived very well, and there's no aggregation of microglia and no signs of activation looking at the CD68 labeling. So really, what we see is a picture of microglia inactivation or not responding to injury in a hibernating condition. Whether this is related to hibernation and how this is related to hibernation, what was the mechanism during hibernation such that microglia is not responding to injury, that's something we wanted to know next. And so through literature search, one idea came to us that is immune metabolic regulation. Some of the work you probably know much better than I do by Luke O'Neill and others showing that succinate is an inflammatory signal that induces the inflammatory cytokine through HIF1 alpha. We happened to look at the HIF1 alpha for a different reason, but we found that HIF1 alpha actually is extremely low during hibernation, and this is from retina samples. It's kind of counterintuitive in a sense that you would imagine perhaps during hibernation, you don't breathe much, there's not a lot of oxygen, you're under the animals in a hypoxic condition, and therefore HIF1 alpha may be upregulated, but it's the opposite. You see almost very little HIF1 alpha. And accordingly, when we measure the inflammatory cytokine, there's much lower IL-1 alpha, IL-1 beta in the hibernating retina, indicating perhaps hibernation leads to a somewhat immune suppressive microenvironment. Indeed, when we look at the RNA-seq pathway analysis, we saw that the cytokine receptor activity, immune receptor activity are downregulated during hibernation. But you could say this is all global retina tissue, nothing really pinpoint to microglia. So to this end, we performed single cell sequencing. As you can see that retina are notorious for having many types of neurons. But we were able to identify the microglia from the data set, and we were able to compare the awake versus hibernating condition. As you can see that for microglia, we know that a lot of the mitochondrial genes are downregulated, and some of the genes such as P12Y12 usually indicative of more homeostatic states of microglia upregulated during hibernation. But when we look at the neuron, the picture is quite different. In fact, a lot of mitochondrial genes are upregulated. So overall, when we compare, here's more examples of genes, molecules associated with succinate dehydrogenase, DCA cycle, they are completely oppositely regulated among neurons versus microglia. And pathway analysis showing the same that a lot of pathways are downregulated in microglia but upregulated in neurons, suggesting that there are a differential regulation metabolically for neurons versus non-neuronal cells. And more detailed analysis of microglia indicating that in the hibernating condition, they become more ramified compared to the awake ones. So perhaps we can see immune metabolic regulation during hibernation that render the microglia to be less active, not responding to injury. But is that related to succinate dehydrogenase suppression? And for that, we look into the literature again, and we identify some of the potential links. For instance, this work, again by Luca Neal's group, and identify that succinate dehydrogenase, being a key regulator, sort of a switching macrophage from inflammatory, pro-inflammatory phenotype to anti-inflammatory phenotype. When there's an infection, glycolysis increases, mitochondrial memory potential increases, and succinate oxidation increases. And along with this, there's a huge generation of mitochondria ROS, which leads to the HIF1 alpha and ultimately inflammatory cytokine. So we imagine during neuroinjury there could be let me see, there could be increased glycolysis and increased succinate oxidation and mitochondrial rust production, and which through HIF1 alpha regulation to ultimately leads to more cytokine production. But as I have already shown you during hibernation, a lot of those are downregulated. The glycolysis, succinate oxidation, I show you the much lower HIF1 alpha and much lower inflammatory cytokine. So everything seems to go against the reactive microglia response to injury, except that the most critical molecular underpinning that is the mitochondrial rust, which we don't know. And I think it's very important information we need to probe. Again, coming back to our previous study looking at the co-resistance or co-adaptation, one of the key mechanisms we identified, in fact, is the mitochondrial rust overproduction in the non-hibernating cells, but hibernator appeared to be able to avoid that. So we really wanted to measure the mitochondrial rust in the situation of injury and in hibernation. So we developed a method which is quite complicated because it's not easy to measure mitochondrial rust. And because if you want to take the sample, you have to cut the optic nerve to take the retina, and that's an acute injury, could well lead to acute upregulation, a surge of rust. To avoid that, we developed this method. We would do a partial optic nerve crush, and then 3 days later, we will perfuse the animal with fixative so that we can preserve the molecules in fix the condition. But before we did that, 3 hours before we did that, we injected mitochondria-specific ROS sensor called mitoneal D. And 3 hours later, we perfused the animal. And then we will take out the retina and we will image the retina right away to look at the mitoneal D signal. And then we would process the tissue for immunolabeling. And then we image again to identify the same location so that we can looking at the same cell. So here's an example of what we got for the awake animal. And this is the injury site, and you can see very significant upregulation in mitochondrial signal compared to the uninjured site. But this is much muted in the hibernating animal with the same injury. Now, if we zoom in, you can see that there's lots of red dots on the injury side that's not presenting in the uninjured side. If we zoom further in, those are in fact mitochondrial signals, and those are later shown with IVO1 positive staining in an IVO1 positive cell, but not GFAP positive cells. So they're microglia or macrophage indeed, but not astrocytes nearby. And we also confirm those signals are from mitochondria by co-labeling with mitochondrial proteins such as TOM20. And this is the quantification. And then we use a succinate inhibitor, which is dimethylmalonate, and in the awake animal with the injury, we show that by inhibiting succinate dehydrogenase, we could also very significantly reduce the mitochondrial rust production, confirming that this is related to mitochondrial rust production. So what does the DMM do to microglia? In order to find that, we perform both in vitro and in vivo experiment. In vitro experiment, we use the BV2 microglia cell, and we challenge the cell with LPS, which will lead to inflammatory response, cytokine production, and we show that this suppression of succinate dehydrogenase with DMM can dose dependently reduce the cytokine production in response to LPS. And they also are very specific because it does not change the production of the TNF alpha, for instance. In vivo, when we inject succinate dehydrogenase intraocularly, and after that, we measure the cytokine production in the retina. Again, that we show that both IL-1 alpha beta decrease with the treatment of DMM. So now we have some evidence that this microglia inactivation during hibernation through immune metabolic regulation is likely by succinate dehydrogenase inhibition and through the molecule of mitochondrial rust. Now, the most important question, obviously, and to close the loop, is whether this is responsible for neuroprotection. And to test that, we applied DMM to active animals with the optic nerve injury, and we show that indeed intraocular injection of the DMM in the active animals can also protect the neurons and reduce microglia activation and aggregation and activation. And the activity measurement through MEA confirmed the protection of neurons. And this is the overall protective effect with DMN, and 3 weeks later, in particular 2 weeks, short term has a very good protection. So we think that by manipulating this metabolic pathway in addition to other cells, microglia could be a mechanism to leads to the neuroprotection during neural injury in hibernation. Now, one dirty trick I didn't tell you is that all those application of the DMM was before the injury. So in order to be valuable for clinical application, you really have to show the efficacy of post-injury application. So here, and the other one is that you could argue that ground squirrel is very different from other mammals. They're hibernator, they're weird, special. So here we perform the optic nerve injury in a mouse model and apply DMN after the injury, and we show that neuroprotection of retina ganglion cell after the injury. In addition, to be further closer to the clinical application, we, in collaboration with former NEI fellow Yikui Zhang, now a very successful researcher at Wenzhou Medical University, along with Yang Hu, well-renowned glaucoma researcher at Stanford, we developed this large animal model using goat as a model to have injury of optic nerve not behind eyeball but very close to the chiasm, which is more clinically relevant. In this model, again, we show that intraocular application of DMN can be very protective of the optic nerve injury. So with that, I will just quick summarize what we think that hibernation could insert metabolic regulation into, of course, all the cells, but perhaps in particular in microglia and other possible immune cells, as a result to prevent its overreaction to injury and neuroinflammation, and it leads to a neuroprotective effect. And what we think, what we identified potentially is a metabolic switch, which is perhaps temperature independent mechanism of regulating microglia activity that leads to neuroprotection. And with that, I would like to thank the people in the lab. My apology, this is a little outdated version of the current lab member, but I highlighted people that contribute to this project, and particularly Tan Tai Zhao, a former postdoc, now a physician at Shanghai Second Eye Hospital, is the major contributor, and she's a superb retina surgeon. Xiao Chuan, now at Wuhan Renmin Hospital, was the first to develop the injury model, partial injury model. And Jingxing O has been collaborating with me for a long time, and he contributed a lot to the development of model as well. And then Francis Nadal, Josh, and Vincent, and John Ball both contribute to the work. We have fantastic collaborators, Josh and Luca, from both Cambridge. And Josh was an MD PhD student at NIH Oxford Cambridge program. And Xie Zhi at the Zhongshan University did a lot of bioinformatics with us. And with that, I would be happy to take any questions. Thank you.
Okay. Thank you, Wei. It's always quite amusement and entertainment. So we can start the Q&A session, and then if anyone have questions, please raise your hands or put your question in the chat box. I think I'm gonna start to kick off. A few
Sure.
questions I have. First of all, maybe I missed it, but the injury on the axon in the retina, I'm not clear how this cell-cell interaction modulate the microglia metabolism through cystine. Because I mean, what's the mediator through the axon injury to communicate with the microglia?
Well, that's a really $1 million questions that we, I guess, the field of neuroinjury is still working on. The signal from dying neurons. There's a lot of theories. Obviously, you probably know more than I do. For instance, ATP or other metabolites that will trigger the initial response of injury, or whatever the damaging molecules or signals that triggers the initial response. and there could be other cells involved, exercise, for instance. So that is a complicated, unsolved issue, I'll say.
Okay,
But
so
almost universally, whether it's acute injury or chronic degeneration, this local immune response is always there. But yeah, I don't think it's clear what the signal from the neurons to the glia or immune cells.
so you're saying that regardless the cause, I mean the neuron injury or the other cause of the neuron death all can cause this metabolic dysregulation of the microglia, or if the cause is different, the microglia phenotype will be different too.
Well, regardless the cause, there will be a microglia response to it, and this response must have different phases. Some of them are protective, some of them are detrimental. But what we're trying to say is that their response in some way is dependent upon their internal metabolic regulation, and those are the potential target for making them less detrimental.
I see. Uh, my second question is, what exactly the low temperature, modulate this neuron mediated microglia phenotype? I mean, is this something related back to your FOXO1 machinery, or is something else?
Say that again.
I'm just saying that, you observe this neuron protective phenotype during the hibernation, but my question is, what's the machinery of that? Why the low temperature will do that? Is that something related back to your FOXO1 molecular machinery?
Well, in for individual cells, for instance, neurons, I would imagine, although we didn't look specifically, that FOXO1 and some other molecules, for instance, what is that blocking on that? GTX4, those all could be contributing, and that's related to temperature dependent regulation of molecular cellular mechanism. But what we're trying to do is looking at temperature independent ones that are more directly relevant to metabolic regulation that we could use in addition to low temperature. So temperature does a lot of things, most obviously just through its physically Q10 effect, affecting a lot of things, and other such as the mechanism we have identified molecularly for cold adaptation. But what we're trying to do is something that through in the case of microglia, through tackling the metabolic regulation, independent temperature to perform regulation.
Yeah, but my point is that the your phenotype is somehow observed in the ground squirrel, but the all the let's say it must be something unique on these species because although you're trying to find the temperature independent mechanism, but obviously these neuron injury phenotype will not happen on the mouse or other mammals. So what is the unique part from the ground squirrel?
Well, we extended from the ground squirrel phenotype to the mouse model and other models. That is your protective effect, right? Because that's not related to hibernation. Those animals are not in hibernation or even in torpor. Mouse you can induce into torpor, but we didn't try that. And one of the former postdocs trying to do that in a different lab. But those are awake body temperature at 37 with injury, and then we show protection under those conditions. So those are translating from
But
ground squirrel to
on those condition regarding the DMM treatment.
yes,
Yeah.
not
So
naturally, obviously.
Not what?
not naturally occurring, right? Yeah,
Oh
without the
yeah.
treatment.
No, well, for sure, for sure. So my actually real question is why the ground squirrel actually generates so does such kind of assassinate metabolism?
Well, I don't think they generate that response in anticipation of injury. I think it's just so happened that this kind of a metabolic immune regulation, uh, you know, is in a way that prevented to respond to injury in your inflammation that leads to the that normally will lead to cell death. For instance, those animals with injury after the hibernation, as long as they're hibernating, there will be no cell deaths. But if you wake them up, they I don't know. There will be questions come along, but I will say that when they you awake them from hibernation with the injury, they will still neurons will die afterwards. Except that there's some subtle differences that we observe a very, very sharp boundary between the injury and injury site, indicating that what we are protecting, but what we are protecting is perhaps the secondary, information leads to further cell death. But the physical axonal injury cause cell death is inevitable. It's only temporarily protected while they're hibernating.
I see. Okay, there's a few questions in the chat box. First from Gao Yanqing. Very interesting. One question, how to be sure the causal relation between the neural injury and microglia activation? Would that be possible that in hibernation animals, the injury responses were attenuated in neurons, and that is why microglia were not activated?
That's a very good question, a chicken and egg question. But all I can say is when we manipulate externally this metabolic pathway, we could lead to neuroprotection, less neuronal death. So and that perhaps is the only way I can sort of tangentially answer that. I don't think it's very solidly established causal, but by metabolically regulating microglia, you have neuroprotection.
Okay, the next question from Chen Jiadong. Wonderful talk. What would be the final fate for the neurons that survive nerve injury during hibernation? How long will they remain viable?
That's sort of I anticipated yes, the injured cells will die once the animal wake up from hibernation. Um, because that's physical injury of the axon. That's the a different scientific topic because I don't think you can avoid cell deaths of axonal injuries. So that's what people have been actively researching on. The hibernation can only protect them from the kind of a destinated death temporarily as long as they're in hibernation that there's this metabolic regulation to microglia.
Okay. Next question from Zhenan. So I think he's want to ask a general question. How does hibernation affect the metabolic changes?
Yeah, hugely, right? And so one of the key kind of goal of hibernation is to reduce overall metabolic rate. It's suppressed down to less than 5% of normal metabolic rate. So basically, massive metabolic suppression to down to a point of only critical cell surviving or animal tissue surviving conditions, the very low oxygen consumption, and all that. And that is the hallmark of hibernation.
Okay, so I think I still have one questions regarding your DMM treatment in vivo. So how you see this outcome regarding the neuroprotective phenotype or the retinal phenotype is somehow specific to the microglia in vivo?
Yes, and this is difficult because when you apply that, it applies to all the cells. We can only do some of the control experiment. For instance, we measure the neuronal activity. It doesn't change the neuronal activity, overall retina physiology. So it impact neurons less. In this regard, but how does that affect the neuronal intrinsically? That's something we need to further explore. So
So,
at least the drug is non-specific.
so
Now,
what
if
if,
you really want, yeah, go ahead.
well, I just said, what if you deplete the microglia?
Yeah, that's another interesting question, and that we saw some species differences. In the ground squirrel, we saw depleting of microglia have been very protective, but apparently not in mouse situation. So I think again that this is not as simple as black and white. They come activated at the different phase and space and time, and it could be protective or detrimental. And then there could be species difference. So here, the ground squirrel is unique and different.
But when you say
On the
depletion,
flip side, yeah, I do not know mouse is the best model or really close to the human or situation. All I can say is different from a mouse.
no, I just say you say the mouse is opposite to the ground squirrel.
The mouse, you can deplete microglia. It does not to be protective for the retina ganglion cell death in this model.
Yeah, that's my question. So if in the mouse system, because you show that DMM treatment on the mouse as well, right?
Yes, so the point is depletion is never an answer. So
No,
what
no,
you
no.
really,
I
yeah,
know, I know. My question is if you deplete microglia on the mouse and then you treat the mouse with DMM, what happened? Whether this will be somehow improvement on the
uh,
depleted
we
microglia
haven't done
model.
that. We haven't done that. So you're trying to looking at the DMM effect not on microglia but in neurons. Is
No,
that
I'm
what you?
not saying not on micro. I'm just saying that if something this DMM have um, the neuroprotective effect through the
Independent.
yeah, yeah.
We haven't done that. We haven't done.
Okay. Okay. Um okay, so I do not see any further question in the chat box. But I think it's truly inspiring and then very interesting. I think anyone from the audience have questions, please reach out to Wei. If you have other hibernating related questions. But if you do not, if no one actually have further questions, thank you very much, Wei, to give us this lecture, and then I really appreciate it.
Thank you, Tran.
Yeah,
Always be
yeah.
happy to be back as a speaker or host anytime.
Will do. Okay. Okay.
Take
Thank you,
care.
everyone.
Bye.
Okay. Bye-bye. Bye.